the phosphate group must come together with this structure. The phosphate group comes like this is P, this is OH, double bond O, OH and OH here. Now see, these two hydroxyl groups are close together and these two hydroxyl groups will condense now and it will remove one water molecule. After removing one water molecule, the structure will become Hello Bandhugan, Namaskar and welcome back to my YouTube channel Sahil Educations, and today we'll be discussing about the DNA. So first let us know that what is DNA. It is a polynucleotide basically. So polynucleotide means there are a lot of nucleotides are going to combine together and they are going to make a double helical structure, and that double helical structure is basically we call DNA. Now what do this DNA do in our body? They perform as a genetic material in a cell. A cell contains a lot amount of DNA and that DNA can be considered as the genetic material. And the full form of DNA is, as written here, deoxyribonucleic acid. So if we elaborate this written full form, if we talk about the full name of DNA, then there is D, then oxy, and then ribo, and then nucleic acid. So D means devoid of oxygen in the ribose sugar, and when it is present in the nucleus and the nature is acidic due to the presence of phosphoric acid, it is nucleic acid. So you have heard that DNA is present inside the nucleus, and in case of bacteria or in case of prokaryotic cells, the DNA is present in the cytoplasm everywhere. Wherever you just pull out the cytoplasm, you will get some amount of DNA. Okay, so this DNA has, if we talk about the DNA in more details, then we will know about the nucleotide and nucleoside. So what is nucleotide basically? As we know that the DNA is polynucleotide, so we have to know about the nucleotide. So nucleotide is a molecule which is made up of, or which is actually produced by the mixture of ribose sugar, nitrogen base and the phosphate group. Whereas if we talk about nucleoside, which is very similar with the nucleotide, then nucleoside is made up of ribose sugar and nitrogen bases. So there is no phosphate group in case of nucleoside, and one phosphate group is obviously present in case of nucleotide. So let us know about the ribose sugar. So this one is the structure of ribose sugar. It is a 5-carbon compound, and when the compound is deoxy, then you have to know that there is one oxygen less in this structure. So you have to remove one oxygen from the carbon number 2. That is why this ribose sugar can be called as 2-deoxyribose. Okay. Apart from this, now talking about the phosphate group, from where this phosphate group or from where this phosphate coming, and if we want to know about this, then we have to know the structure of phosphoric acid, which is actually providing the phosphate in this vast molecule. So phosphoric acid has a structure like this: P double bond O, OH, OH and OH in single bonds. Now talking about the nitrogen bases, so nitrogen bases are basically of two types. One is purine and another one is pyrimidine. So purine bases are again of two kinds. One is adenine and guanine. The structures of adenine and guanine is as shown here. There is actually two cycles, heterocyclic compound, and in case of pyrimidine, these structures are more simpler than the purines. So pyrimidine has three parts or three types. One is cytosine, thymine and uracil. So this structure resembles the thymine, this is cytosine, and this one is for uracil. Uracil is actually present in case of RNA. This uracil is not present, is never present in case of DNA. Okay. Let us know about the polymerization, or let us know about the production, or let us know about the making of basically this nucleotide, how this nucleotide looks like, how this is formed. So we have to go through this one. So nucleotide, we know that nucleotide contains ribose sugar, then phosphate group, and the nitrogen base. Whether the nitrogen base is adenine guanine or cytosine thymine. So it is not a matter. So matter is how they are combined together, how they are formed. So let's see. Just draw ribose sugar here, a 5-carbon compound, and we have seen that the ribose sugar, whatever is going to make DNA, is to be deoxyribo. That means number 2 carbon must devoid of oxygen. So this one is OH, H, H, OH, H, OH, and here H and CH2OH. Okay, so this is your ribose sugar, and you have to remove the oxygen from carbon number 2, as this one is carbon number 1, this 2, 3, 4 and 5. So next you have to add the nitrogen base, whether the nitrogen base is adenine guanine or cytosine thymine. So always remember that the nitrogen base must be added in the carbon number 1 with the carbon number 1, and then the phosphate group must come together with this structure. The phosphate group comes like this is P, this is OH, double bond O, OH and OH here. Now see, these two hydroxyl groups are close together, and these two hydroxyl groups will condense now, and it will remove one water molecule. After removing one water molecule, the structure will become so. This is the structure what you get after combining the nitrogen base, phosphate and ribose sugar all together. So this one is your actually nucleotide. Now if we talk about the direction of this nucleotide, we know that this one is the 5th carbon. So this direction, this part is the 5 prime of the nucleotide, and this one is the third carbon, number 3 carbon. So this part is the 3 prime of the nucleotide. Now we would know, now we are going to know that how these nucleotides are going to combine with each other to make polynucleotide. So let's see how two nucleotides are combining each other. Is looking like double bond O, single bond OH, single bond O, then it is CH2, then H, and then here the ribose sugar is present. Suppose adenine was there, and a free hydroxyl group was present at the 3rd carbon of the ribose sugar. Then another compound, another nucleotide is coming here, and it will bind with the nucleotide before. So it will look like OH, P, double bond O, single bond OH, then O, and then CH2, then H, and another ribose sugar is present here along with the free hydroxyl group at the 3rd position, and at the first position, suppose guanine is the nitrogen base. So see, two hydroxyl groups are close together, and they will again condense and make a structure like this after removing one water molecule. So the structure will be looking like OH, P, double bond O, single bond OH, then O, then CH2, then H, and along with this you will have the ribose sugar with nitrogen base A, and here one oxygen is remaining now after removing the H2O. So it will be O, then P, double bond O, single bond OH, and then O, then CH2, then H, and again the third carbon will have the free hydroxyl group along with the guanine as nitrogen base in the first carbon of this ribose sugar. So if we talk about, this one was the molecule number 2 and this one is the molecule number 1, so both molecules have made a bond together, and now the bonds whatever has made here is phosphodiester bond. That means the phosphate is present in between, there is a ester bond, there is an ester bond, and another ester bond is present at this region. So two nucleotides are combining together with the help of phosphodiester bond. So phosphodiester bond is actually going to play a role as a backbone of your DNA helix or DNA molecule. So the backbone of your DNA is made up of phosphodiester bond, and actually is going to help to make the backbone, and the backbone is made up of ribose sugar, whatever. Now talk about the opposite side. So if we talk about the opposite side, let's see the direction of this direction of this polynucleotide actually. So this one would be the 5' direction as the fifth carbon is here, and this one is going to the 3' direction as the third number carbon is here. And now see the direction and the strand in the opposite direction. So suppose A is going to bind with T, we all know about this, so T is here, and now so this one is the first carbon, this one is the second, this one is the third, fourth and fifth. Okay, so this third carbon has OH free here, and you will see so this must contain C here, and this one is going to be the fourth carbon, and here the fifth carbon. So fifth carbon must contain a phosphate group along with this, so P, double bond O, single bond OH, single bond OH here. So this one is H, and this one is going to be the third carbon. So along with this third carbon you will have oxygen, you will have phosphate, you will have oxygen, and then you will be going to direct with the fifth carbon of another ribose sugar. So fifth carbon of another ribose sugar will look like CH2H, and here this, and this carbon must contain T, that is why A can bind with T, and the third carbon of this ribose will have free hydroxyl group. So the 5' is in this part and 3' is in this part. And the two opposite strands, the bonds are present in between the nitrogen bases. Basically the nitrogen bases, which is A bonding with T with double bond and G bonding with C, must contain 3 hydrogen bonds. So these are basically the hydrogen bonds. If you are going to be asked that what kind of bonds are present in DNA, then DNA must contain phosphodiester bond and hydrogen bond. If you are going to be asked as what is the bond which is going to make the backbone of DNA, then you must say that the phosphodiester bond is the only bond which is going to help making the backbone of a DNA. So that's all for today. Thank you very much. See you in the next video.